The first thing that affects glycolysis is transportation of glucose by a GLUT transporter. We have 5 types of GLUT transporters and they are divided into 2 groups: insulin-dependent and insulin-independent. The only insulin-dependent transporter is GLUT4. GLUT4 transporter primarily located in adipose tissue and skeletal muscle tissue, and GLUT4 transporter called insulin-dependent because insulin can increase the amount of GLUT4 transporter on the cellular membrane of adipose and muscle cells. And the more glucose transporter is on plasma membrane, the more glucose molecules enter per unit time, so the higher will be glucose income into the cell. Glycolysis itself is regulated on 2 stages. On the initial stage, the rate of glycolysis is regulated by hexokinase and glucokinase. Hexokinase is inhibited by glucose 6-phosphate, which is the product of the reaction that is catalyzed by hexokinase, and also important that hexokinase cannot be stimulated by hormones, so insulin have no effect on hexokinase. In contrast to hexokinase, glucokinase has a completely different features. First of all, glucokinase is not inhibited by glucose 6-phosphate, and which is even more important, insulin can increase the speed of glucokinase. So glucokinase has hormonal regulation, which is provided by insulin, that stimulates this enzyme. The major regulatory enzyme of glycolysis is phosphofructokinase. Basically, this phosphofructokinase regulation affects both glycolysis and gluconeogenesis. Activation of phosphofructokinase stimulates glycolysis and simultaneously inhibits gluconeogenesis. Such regulation called reciprocal regulation. In context of glycolysis, we will discuss just the effect of this enzyme on glycolysis. In glycolysis, fructose 6-phosphate is converted by phosphofructokinase 1 into fructose 1,6-bisphosphate. The activity of PFK1 is regulated by the concentration of fructose 2,6-bisphosphate. Increase in amount of fructose 2,6-bisphosphate increases the activity of PFK1, and the concentration of fructose 2,6-bisphosphate is regulated by 2 enzymes. The first one is phosphofructokinase 2. PFK2 converts small amount of fructose 6-phosphate into fructose 2,6-bisphosphate. As a result, the activity of fructose 2,6-bisphosphate increases. And also the second enzyme that regulates fructose 2,6-bisphosphate level is fructose bisphosphatase 2, FBPase2. FBPase2 degrades fructose 2,6-bisphosphate into fructose 6-phosphate. So FBPase2 decreases the level of fructose 2,6-bisphosphate. Actually, PFK2 and FBPase2 are 2 different domains of one enzyme. So because domains are different, to simplify we can imagine that it's 2 separate enzymes. So we see that PFK2 increases fructose 2,6-bisphosphate level and FBPase2 decreases fructose 2,6-bisphosphate level. And the key concept here is that the activity of both these regulatory enzymes is controlled by hormones, insulin and glucagon. Insulin stimulates the activity of PFK2 and simultaneously inhibits the activity of FBPase2. So production of fructose 2,6-bisphosphate increase and degradation decrease. So the level of fructose 2,6-bisphosphate increase. And increase in the level of fructose 2,6-bisphosphate stimulates the activity of PFK1 that converts fructose 6-phosphate into fructose 1,6-bisphosphate, thereby insulin activates glycolysis. In contrast to insulin, glucagon stimulates the activity of FBPase2 and simultaneously inhibits PFK2. So in this case, degradation of fructose 2,6-bisphosphate prevails over production. This results in decrease in fructose 2,6-bisphosphate level, and decrease in fructose 2,6-bisphosphate level inhibits the activity of PFK1, thereby conversion of fructose 6-phosphate into fructose 1,6-bisphosphate decrease. Thereby glucagon inhibits glycolysis. So insulin stimulates phosphofructokinase 1, thereby insulin stimulates glycolysis. Glucagon inhibits phosphofructokinase 1, thereby glucagon inhibits glycolysis. Also PFK1 has allosteric regulation. The logic here is that during some energy consuming process, let's say for example during muscle work, ATP is consumed for muscle contractions. ATP is degraded into ADP with release of energy, and then ADP is degraded to AMP. So basically increase in AMP level means that tissue is doing some energy consuming work, thereby tissue requires additional energy. and additional energy can be produced by glycolysis. So because of that, AMP stimulates PFK1, thereby stimulating glycolysis. In contrast to this, if tissue have a lot of ATP molecules, it's a sign that tissue currently is not doing substantially energy-consuming work. Thereby tissue do not require additional energy production, so because of that, ATP molecules inhibit PFK1 activity. And much wiser in such energy-saturated state to prepare some energy reserves. This PFK1 inhibition eventually causes accumulation of glucose 6-phosphate, and glucose 6-phosphate can be converted into glucose 1-phosphate, and glucose 1-phosphate can be used for glycogen synthesis. So tissue can prepare some energy reserves in form of carbohydrates. But very important that glycogen stores are limited, because glycogen is not the optimal material to store. The reason is that glycogen is basically a complex of glucose molecules, and glucose is a hydrophilic molecule. This means that glucose attracts water. So when tissue stores glucose molecules in form of glycogen, this glucose liver also stores water. And actually it takes a lot of space to store both glucose and water at the same time. The optimal storage material in our body are triacylglycerols. Because they are totally hydrophobic, they do not attract water, and because of that they are very compact substances to store. Triacylglycerol molecule is basically 3 fatty acids connected to glycerol. So when tissue stores triacylglycerols, tissue stores glycerol that can be used for energy production, and also 3 fatty acids that also can undergo beta oxidation with energy production. And the substrate for fatty acids production is acetyl-CoA. So to store energy in form of triacylglycerols, organisms have to produce from glucose acetyl-CoA, and then to use acetyl-CoA for fatty acid synthesis. So how this process occurs? Eventually glycolysis results in production of pyruvate, and pyruvate is transported into the mitochondria, where pyruvate dehydrogenase converts pyruvate into the acetyl-CoA. Acetyl-CoA binds to oxaloacetate with formation of a citrate, which is an intermediate metabolite of the Krebs cycle. If tissue requires energy, then citrate is going through the Krebs cycle, where dehydrogenases pluck out electrons out of acetyl-CoA molecule, and this results in formation of NADH and FADH molecules. And very important that one of the dehydrogenases in the Krebs cycle, called isocitrate dehydrogenase. And this NADH and FADH electron carriers deliver electrons to electron transfer chain that results in energy production. But at some point, the amount of ATP becomes sufficiently high, and tissue do not require additional energy. What tissue requires is to create energy stores in form of triacylglycerols. And triacylglycerols are stored in the cytosol. To make triacylglycerols, tissue have to produce fatty acids. Fatty acid synthesis also occurs in the cytosol, and the substrate for fatty acid synthesis is acetyl-CoA. So somehow acetyl-CoA have to be delivered from mitochondrial matrix into the cytosol. And the problem is that mitochondrial membranes do not have any transporters to deliver acetyl-CoA from mitochondrial matrix into the cytosol. So acetyl-CoA cannot cross through mitochondrial membrane. But the substance that can cross through mitochondrial membrane is citrate, because mitochondria have so-called citrate shuttle, and citrate, through citrate shuttle, leaves mitochondrial matrix and enters into the cytosol. In the cytosol, citrate is cleaved into oxaloacetate and acetyl-CoA, and now acetyl-CoA can be used in fatty acid synthesis. So when tissue becomes energy saturated, high amount of ATP molecules inhibit isocitrate dehydrogenase enzyme in the Krebs cycle, and inhibition of the Krebs cycle eventually causes accumulation of citrate in the mitochondrial matrix. And citrate from the mitochondria is transported into the cytosol, where citrate is cleaved with release of acetyl-CoA that can be used for fatty acid synthesis. So as we see, if the amount of ATP is high, then the amount of citrate is high. So high citrate level in the mitochondria and in the cytosol is the marker of energy saturated state. And in energy saturated state, tissue do not want to produce additional energy. So increase in citrate level inhibits PFK1, thereby inhibiting glycolysis. Because the purpose of glycolysis is to produce energy, and we have already energy sufficient state. However, if increase in ATP level and increase in the citrate level inhibits PFK1, with PFK1 inhibition glycolysis decrease, and thereby acetyl-CoA production decrease. And as we know, fatty acids are produced from acetyl-CoA, so with decrease in amount of acetyl-CoA, fatty acids production have to decrease. So if energy saturated state leads to decrease in fatty acids production, then how liver tissue can produce massive amount of fatty acids in energy saturated state? How liver tissue can create these massive triacylglycerol stores? The reason is that, as we said, the major regulatory enzyme in glycolysis is PFK1, and the major factor that regulates activity of PFK1 is PFK2. And the key concept here is that the activity of PFK2 is stimulated by insulin. And when we consume a cake, for example, glucose level in the blood increases, and this provokes pancreas to secrete insulin. And increase in insulin level stimulates PFK2, and PFK2 subsequently stimulates PFK1, thereby stimulating glycolysis. And very important that PFK2 stimulation basically overrides any inhibition by ATP or citrate, at least in the liver tissue. So the whole concept here is that in the liver tissue, liver subtype of PFK2 strongly depends on insulin level. And if insulin level in the blood increases, this stimulates PFK2 that subsequently overrides inhibitory effects of ATP and citrate molecules, and this stimulates PFK1. And this permits liver tissue to degrade glucose molecules into the pyruvate and subsequently into acetyl-CoA. And now because high ATP level inhibits Krebs cycle, acetyl-CoA can be transported in form of citrate into the cytosol, and in the cytosol, acetyl-CoA are used for fatty acid synthesis, and this permits liver tissue to create triacylglycerol stores. And also fructose has the highest metabolic rate in liver tissue compared to any other monosaccharide. The reason why liver metabolizes fructose faster than any other monosaccharide is that fructose is the only monosaccharide whose metabolic pathway bypasses PFK1. As we know, in glycolysis, the major rate-limiting enzyme is PFK1. This enzyme controls glycolysis by regulation of conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. And recall that the final product of fructose degradation is glyceraldehyde 3-phosphate. It's the intermediate metabolite of glycolysis that enters into glycolysis after PFK1 rate-limiting point. So the final product of fructose metabolism, glyceraldehyde 3-phosphate, do not inhibit phosphofructokinase 1, and this means that liver metabolizes fructose without PFK1 inhibitory effect. Because in contrast to this, if we take glucose or galactose, they both enter into glycolysis through conversion to glucose 6-phosphate, that then is converted into fructose 6-phosphate. So they both enter into glycolysis prior to PFK1 rate-limiting reaction. So it's the reason why fructose is metabolized by the liver tissue faster than any other monosaccharide.